So it's really nice to be here, and actually my career in plant sciences started in York, just down the road in the University of York, so it's nice to be here, kind of back where I first got interested in plants. So when I was asked to give this talk, I was told, oh, can you talk something about curiosity, your curiosity in plant sciences? So I thought, what do I actually spend my time doing? And I kind of spend my time looking at some pretty pictures and thinking a lot about one particular graph. So I realise I'm perhaps not selling this, great, but if you stick with me, hopefully I can kind of convince you that this is interesting. So for this talk, we have to go right back to the beginning. So we go back to this primordial soup, which over time, starts to organise, and we get macromolecules, and importantly, we get membranes. And once you have membranes, then you can have organisation. And for me, an important part of this is that it means that these protocells have now got a size. They've got a defined size, and that means that the components inside them have got defined concentrations. And when you have concentrations, interesting kind of dynamics can happen with chemical reactions and things. And so if we actually kind of then think about cells and what size they need to be, we can think about some kind of ancestral eukaryotic ancestor, and its optimum size is going to depend on how well molecules can diffuse through the cell. It's going to depend on how the size of the cell affects its biosynthetic capacity. Can it make enough energy to survive? And whether it can build the right intracellular structures that it needs, for example, to be able to divide. And the optimum size is going to change. It's going to vary between species, but it's also going to vary within species, okay? So if there are environmental changes, it's got to change. And so there has to be like dynamic adaptation of cell size. If then we kind of go forwards through evolution a bit more and we start to think about multicellular organisms, all of these things I've just talked about still matter to those cells within the organisms, but we now also have these other aspects that happen at higher levels of organisation. So the cell size is going to affect tissue area, it's going to affect tissue structure, and it's going to affect specialised cell types. And one thing that I'm particularly interested in is whether this dynamic control, which is evolving for environmental control, is actually being co-opted by development to be able to build different structures. So actually where I started looking at this is in the meristem. So here's an Arabidopsis plant, and this is the shoot apical meristem. So we cut off the tip, we remove with forceps all the buds that you can see there, and eventually you get to this structure. This is a confocal image, so we've taken a stack of the structure and then we've projected it into what we call 2.5D. So we're keeping some of the shape, but we're just looking at the outside layer of cells. And so these are the stem cells right in the middle of the structure, and these are the developing organs towards the outside. And if we take this image, we can do some analysis, and we can look at the size of the cells here. So we have the stem cells in the middle. When we look at them, we can see there's a really tight distribution of cell sizes. And the interesting thing about this is this structure, even though we've got this static image, is really dynamic. All the cells in that are constantly growing, which is pushing them towards larger sizes, and they're constantly dividing, which is taking the largest cells and putting them back into the middle of the distribution. And we know that in unicellular organisms, the balance between growth and division happens at the level of individual cells. But we thought, well, here we've got this structure where evolution has developed these mechanisms to coordinate what the cells are doing. Do they still need that individual control, or is development actually taking that part away? So what I did was to take time-lapse movies of the shoot apical meristem growing and identify all the different cells in it and track their lineages back. So we got loads of data from this, and we could find out like how much variation actually is there in cell size. But that on its own was quite hard to understand. So we decided to try and use some mathematical modelling to help us understand it. So this is a really, really simple model. It's really just a computer simulation, and it works basically on logic. So we've got two loops going on here. You've got a growth loop here. So each time step, the cell grows a little bit according to this growth rate, which changes its size. And then on this right-hand side, we have a cell cycle. So basically each time step, the cell produces some CDK activity. CDK is what drives the cell cycle forwards. And we check the concentration of CDK activity against a threshold. If it's not met the threshold, it goes round again. We do more growth and we do more CDK production. If it has met it, then the cell divides and it gets smaller. So you can see in this hypothesis, we're just testing that these two things kind of happen independently, because the only link that we've got between them is when the cell divides. So this is just working as a clock on this side that every so often is going to tell the cell to divide. And when we simulate this based on the variation in growth rate and the variation in division ratio that we saw in our time lapse, we can see that we started with a very narrow distribution, but over time this is getting wider and wider, and this is because the cell lineages, some of them get bigger and bigger and bigger each time step, and others get smaller and smaller and smaller as they go on. So we adjusted this and said, well, what happens if we put in another link across these two loops? And you can see now what happens is that the distribution of cell sizes stays quite constant. There's still some variation, but it's remaining stable over time. So this kind of shows us that this would be a possible mechanism, and this would be more of an active control of cell size, but we needed to test it. And this is where I get to introduce the graph, the dull graph that I like to look at. And so what you're seeing here is the simulated cells, the size that they were when they were born, and then how long it took them to divide. So this was important because it was something that we could actually test in our experimental data to see if our model is doing something relevant. But it also explains why this works. Because if you think about it, if you have a cell that's large, according to this graph, it's going to divide after a short amount of time, which means it's not going to have time to double completely before it divides, which means instead of its daughters coming out at the same size, they're going to be thrown back into the middle of this graph. If you, on the other hand, have a small cell, it's going to take a long time to divide, which means that it more than doubles, which means its daughters are going to be larger. So you have this kind of sorting that's going on in this, kind of as an emergent property, yeah, where the extremes are getting sorted back into the middle. So we looked in our experimental data, and it's kind of, you've got lines. That's because of the time points that we take, but you can see that we do have this kind of inverse relationship. And then when we looked at individual lineages, we could also see this and prove to ourselves that this is kind of what's happening. So here you have two cell lineages, one that divides symmetrically, so its daughters are very similarly sized, and you can see that they both divide after about the same amount of time. On the other hand, here we have two cells that are different sizes when they were born, and the one that's smaller takes longer to divide, and it's kind of catching up with the larger cell before it divides. So we can see that we've got some kind of active correction of cell size, despite all of the signals that we've got for development that are also controlling the plants. So we've been looking into this in different ways to try and understand this mechanism, but I wanted to kind of talk about one thing that bothered me, which is, well, we've got this mechanism to keep cells the same size, but during development, you don't want to keep cells the same size. You want to let some cells get big, and you want to let some cells get small. So if we had, for example, cell control in the leaf epidermis, would it actually be counterproductive and working against what we want to happen? Because we want to get these large pavement cells, and we want to get the small guard cells to make the stomata. So we decided to look at the stomatal lineage, and these are the divisions that it undergoes. So you start with a protodermal cell, and this kind of enters the lineage and takes on this specialized identity. And then it undergoes several asymmetric divisions, where the cells are different sizes when they're born, and they also have different identities. So this one marked in yellow is the one that's going to keep dividing. And then after a certain amount of time, there's another fate switch, and some more transcription factors are turned on, which produces a final symmetric division. So we can see this in the movie that we've made. So here you're seeing in green is a plasma membrane marker. In yellow is a marker that we've made for cell cycle progression. So it basically comes on when the cells start replicating DNA. And then there's also a blue marker, which is kind of hard to see. But if you do see it, it's for this transcription factor here. So it's basically just marking the very last stage of the differentiation. So hopefully this plays. And if we look at this lineage, you can see that there are asymmetric divisions going on. I'll just play it again. Here, so it divides asymmetrically and again, and now this one is the last symmetric division that's given the two guard cells, and those are going to differentiate more to make the stomatal pore. And the interesting thing in this for me is that these asymmetric divisions reverse the rule that we saw in the shoot apical meristem. So in the meristem, cells that are large divide quickly. In this situation, cells that are small divide quickly. So we wanted to kind of know, does this destroy cell size control? So first, actually, we decided to look at this last symmetric division. And I just thought I would ask whether people would like to make a prediction, whether you want to raise your hand if you think there is cell size control in this division. And that there isn't. Okay, so it's a great prediction. So we actually thought it was possible that there wouldn't be, and that's because this transcription factor here controls the expression of loads of cell cycle genes. So we thought when this came on, it might just sort of flood the cells and stop them being able to sense size properly. So you can see here, this is that neat transcription factor coming on, and that comes on in the cells a certain amount of time after they're born, whatever size they are. It takes that long to come on. So that's kind of like the timer. But when we look at the G1 to S transition, so that's the first step of the cell cycle, you can see that there is cell size control here. So that kind of makes sense. It's a symmetric division like in the meristem. So that's great. So then we looked at the asymmetric division, and again, well, I'll ask if people think that there is cell size control, or if there is not cell size control. Okay, so some votes for no cell size control. Well, so actually, you get to be right and also wrong, because this is actually my new favourite graph, because what you can see here is actually you get a bit of both. So we get this portion of the graph where we have a straight line, it's acting like a timer, and this portion of the graph where we're getting cell size control. And what we know is that there's not really any further changes to the genes and things at this point. There's no changes to the network, it's just the behaviour that's changing. So the cells come in and are big, they divide really quickly following a timer, and then they go into this phase that when they're getting closer to this point where they're going to make the last division, where they start showing cell size control, which I think is really interesting and kind of links to that question of whether some of these environmental pathways might be being used for development. So we're kind of using that original model to try and understand how this happens. I won't really kind of go into it, but we can show how each of the changes to the cell division rules contributes to producing the different shape of the graph. So this is my group, and just to kind of like point out that even within a group, people are doing all kinds of different things, and that's one of, like, I think the fun things of working together, because we share ideas and come up with new things. So Ash is working on some really detailed physiology experiments. Matt is using some really complicated physics techniques. Billy is a geneticist, and he was looking at fluorescent proteins and quantifying them in cells. And Emma did various things, but she was the one who produced the stomatal data. We're always interested to hear from people if you would like to come and do some work experience or something in the lab. Thanks.